So what happens when two paramecium come together like this, and then they have a macronucleus, macronucleus, micronucleus, micronucleus, right? They come together, and then after that, we quickly just making the long story short, and I don't expect you to know all of the details. But what happens, the macronucleus disappear, the micronucleus undergo meiosis 1, and then meiosis 2, right? You remember in meiosis, you start out with 1 cell, at the end you have 4 cells. Here you have started out with 1 nucleus, and at the end you have 4 micronucleus, remember? The micronucleus disappears, making the long story short, and then 3 of these guys disappears, right? And then 1 of them undergo mitosis, becomes 2, and then what happens, this one goes to here, that one goes to there. All of those details, but you should know that meiosis occur here. The micronucleus undergo meiosis. That's one thing you should know. You should know micronucleus undergo mitosis as well too. I hope I'm making some sense. Then you should know macronucleus disappear. It will reappear. Okay, so when 1 micronucleus goes to this paramecium, 1 micronucleus goes to that paramecium, then they separate, right? Macronucleus disappear, then you have 1 micronucleus, and then they undergo binary fission, which you see it in the lab. Something like this, you're seeing it in the lab. And then the binary fission is pretty much mitosis. I hope I'm making some sense. So that's one way of multiplication. The other one, when you have the paramecium here, macronucleus, they undergo binary fission, becomes 2, a cell divides to 2, but that's what we have to the board, that's what we have in the lab on the microscope, and that's pretty much it. Is that me? Or is it you guys? It's me.
Yes.
I guess my boss is texting me a meeting
Let's cancel that.
So, okay. So those are how I make, and then what else? So you have binary fission, conjugation. I talked about that. Then, in case of Protista, you also have sexual reproduction. This is not referred to as sexual reproduction for whatever reason. Sexual reproduction, you have microgamete and macrogamete, right? They come together, right? Fertilization. And then you have the oocyst. Out of the fertilization, you have the oocyst. It's not a cyst. In this case, sometimes organisms like Euglena, what happens, you have the flagellated Euglena, then undergoes making a cyst. You have these slides in the lab, and then they come out of encystment, they come out of excisment, and then you have that one. Again, that is just encystment and excisment, but the type of division that is occurring from here to here, this is a cyst. Okay, this is a trophozoite stage. It's a trophozoite stage, and that would be mitosis, right? So they, Euglena undergo binary fission. So what would be an example of here? I gave you, this is paramecium. What would be an example here? Yeah, you know many of them. You should know many of them. No, Giardia does not make microgamete. Giardia pretty much undergo binary fission. Plasmodium. Yeah. All of your apicomplexa. Plasmodium. Who else? Cryptosporidium. Who else? Toxoplasma gondii. Who else? Imeria. I saw a whole bunch of them. Yes, Ruben.
Do you know what schizogony and sporogony are?
Well, I knew it, but for your exam on Tuesday, I will forget it. But since I remember it today, let me go over it. You never know what human memory is very selective, isn't it?
Yeah.
Like we choose who to remember and we choose who not to remember. Like you guys will see me after semester is over. Is that some of you still see me in the hallway or mall? You just turn around that way. Other ones is right? Dylan, do I say it correctly? You're like Dylan, like Bob Dylan. What was his last name? Dylan. Bob was his first name. Dylan was his last name, right?
So,
Yeah. Bob.
Yeah, it was Bob Dylan.
He sang the song.
Yeah, it was Bob Dylan.
What did he sing?
What was it, Hammer song?
Like a Rolling Stone?
Rolling Stone. Oh boy, you guys.
No, that's
No,
the
that was
title of the song.
That's
No, that's the title of the song. Is Like a Rolling Stone is the title of the song.
Oh really?
Yes.
I didn't know
Yes.
that. I thought, doesn't it sing, we're all, no, it's, imagine there was no heaven, is it?
No, that's John Lennon.
Oh, John Lennon, see, I get mixed up too.
Yeah.
You gotta love this class. We talked about blood,
Blood.
feces, bacteria,
Oh,
Bob Dylan.
schizogony. Schizogony. So what happens, you guys? You have a host cell. Remember, imagine this is a host cell. This is the nucleus. I'm going to draw it here. Okay, so what happens? The organism in TER, this is epicomplexo. An example of it. It uses the rope trees and microneedle, dissolve the cell membrane, and then it gets inside of the host cell. So it gets inside. Now what happens, it has a nucleus, right? It will undergo, that cell membrane dissolves, goes away, then undergo several nuclear division. Do that make sense?
Yeah.
And after that, we can talk this to Dr. McRae, say, Dr. McRae, on top of mitosis, we learned something else. This is mitosis, okay? But in mitosis, you also learn that after nuclear division, the cytoplasm divides. Now these guys form the cytoplasm around themselves. That kind of multiplication is called schizogeny. Okay? And then of course, they destroy the cell membrane of the host, destroy the cell of the host, and each one of these guys go and attack, this is the nucleus, attack another host cell, and they keep going and going and going and going. Right? You all know what I'm talking about. Same process, schizogeny in next cell. Now what happens, the other one, well, quickly, some of these guys become microgamete, and some of them become macrogamete, right? They come together and form the oocyst. Oocyst, in some cases that you learn, like in Toxoplasma gondii or Eimeria, they go into the environment, right? In case of malaria, it doesn't go into the environment, okay? So it depends. It depends what we are talking about. Which organism we are talking about, not what we are talking about. Which organism we are talking about. Do I make sense, everybody? Sorry. Okay, so what happens here, then the oocysts, these have one nucleus, these have one nucleus, they come together, still you have one nucleus right there. And then what happens inside of that oocyst, that one nucleus divide, and it depends on the species, you will have 4 sporozoids, they call it. Sporozoid. Okay, and the process, what is happening inside of the oocyst is called sporogamy. Right, that's called sporogamy. What is happening inside of the oocyst, or some people they call it spore. So that's what sporogamy. Some organisms like Toxoplasma gondii, you have 8 of them, right? Eventually, at the end, there are 8 of them. And they stay in the environment until another host ingests them. In case of Plasmodium, they break away from the oocyst, become sporozoites, they find a human, right? And they get transferred by mosquito to human, and in human, you will have schizogeny, and then, of course, a mosquito comes and take a blood meal. Some of those are microgametes, macrogametes. I hope I'm making some sense. You had a few questions. Some of you had a question. When I was talking, I saw your hand. Yes, yes, Nick.
When you go through the order of development for an embryo, from zygote to all those stages,
Okay, sure. Is everybody done with this? Yes.
So schizogeny is when like one cell enters the whole cell and it makes more
Mm-hmm.
cells, and after that, a cytoplasm would wrap around it.
That's pretty much schizogeny.
And then after that, the cells from schizogeny would go into another whole cell, and then
Right. And infect that whole cell. What? You remember I talked about this in case of Giardia, you're supposed to ingest 100 of them in order to become infected? If you, for example, you get one malaria inside of your body, one plasmodium, nothing will happen. You get a few of them, so as a result, they attack our cells and we get sick. You touch the door handle and put it in your mouth, you get one rhinoviruses, you will not catch a cold. But you get a whole bunch of them, you catch a cold. So that's what's happening. They destroy the cells, and then some of those become macrogamy, right? And then in case of Plasmodium, they're ingested by mosquito comes and takes a blood meal. In case of Eimeria, they go outside. In case of Toxoplasma gondii, they go outside. From here to here, it takes 1 to 4 days. Remember that, all of that? You should be able to study your notes over and over and over, and these things come together for you. But looking at it once, I don't think it will come together for you. Yes?
so for the sporogeny to happen, you have schizogeny to happen first for the cells.
Or sporogeny has to happen first in order to schizogeny others, right? It's just, chicken came first.
Do they
or-
both occur in oocyst?
No, what is happening in the
Sporogeny.
oocyst is the process is called sporogeny, and it stops at 8 or 4. Here, it can go on to how many number, I don't know, 20, 30, 50, 100. I'm sure somebody researched that somewhere. I'm afraid to say, look it up in Google.
I have a question. So
It gives you
like,
the wrong file. It gave me yesterday the wrong file. Yes.
example for the, what is it? The phylum of protozoa, would that be like fungi, animals, or plant-like?
Huh?
No, like, what are
No.
the
No,
examples?
phylum. Remember, phylum of protozoa are what did we? Eugleniozoa. We started with Eugleniozoa,
Yeah.
right? And then we ended up with Celiophora. Those are phyla, and I specifically in my notes I said what the name of the phylum.
Yeah.
I said phylum Radiolarian.
Right.
Phylum what?
Radiolarian.
What I think what you're thinking, what happened, Robert Whittaker back
Yeah.
in 1960s, he said that protozoa came from bacteria,
Yeah,
and then protozoa, some of them evolved to animal,
yeah,
some of them evolved to plant,
yeah,
some of them evolved to fungi. I think that's what you meant.
yeah.
Okay. Some of protozoa evolved to fungi.
Okay,
Fungi are not protozoa
all right,
nowadays.
okay.
so fungi share an ancestor with protists?
Yes. Fungi, plants, and animals. Yes.
So our great, et cetera, grandfather was a protist?
Yes.
Okay.
And before that was bacteria.
And then by this,
How do you make up these information?
then the same token, because you can't outgrow your heredity, that is why humans are still animals. Would that argue to mean that humans are still protists?
No.
Let's not go there, please, not today. These guys have exam on Tuesday. Okay, so is there any question here, on there? Yes, yes, yes.
The black part that was in that first box, was that a sporozoa?
No, the sporog and the sporozoites are right here,
Oh,
in the oocyst.
wait, then what's back in the
It was
Those are,
Apicomplexes.
those are schizogeny or schizozoites, sometimes they call it. Sometimes they call it schizozoa,
where did they come from though?
from schizogeny. Right? I draw it on the board. They come from schizogeny, schizozoite. Yes.
How are protista like animals, and how are they not? How are they like plants?
Okay, some of them, because some protista, they do photosynthesis, they have chloroplast, they don't move, some of them. That's why they are plant-like. Some protista, they do move, they ingest their food, just like animals.
Okay.
so I mean on
That's
that diagram, the sporozoites like obviously they like burst out of whatever the
Oocyst.
oocyst is.
Mm.
My question just left my brain.
They will go ahead and infect it. They
Okay.
have to be ingested,
Okay.
and then when they are ingested, they break, like in case of Toxoplasma gondii. When this is ingested, then this wall breaks open in our gut, in our intestine. Each one of them get out, and then infect a cell and become tachyzoites. They call it tachyzoid when they keep multiplying and multiplying very rapidly. Each one of these become tachyzoids in human body. And then our immune system kicks in, slow them down, become bradyzoid. Yes?
When they form the cyst, like the oocyst, that's different from like incest, right?
That's different than, no, this is, okay, good question. So what happens, the process of encystment, he's asking encystment, right? That's what you're asking. Encystment, the process of encystment, the process of fertilization. These are the name of the process. Okay, the process of encystment, that means you have the oocyst, and that oocyst, of course, I said the nuclear division, I erased it in case of euglena, right? The nuclear division does not occur. They form a thick shell. Metabolism slows down, right? All of that is true for encystment. However, in case of apicomplexa, what happens, you have, since this came from a sexual reproduction, the oocyst, same as a cyst, they undergo encystment. They have a thick shell, the oocysts have a thick shell, metabolism slows down. However, the nuclear division continues. And of course, it stops at 8 cells or 4 cells. Do it make sense a little bit?
That's just for apicomplexer, right?
Just for apicomplex. But still, they have a thick shell, the oocysts have a thick shell if they get out of host. If they get out of host. Malaria does not. It gets out of host, but it goes to mosquito. It gets out of host, it goes to human. It gets out of host, goes to mosquito. It gets out of host, goes to human. In case of Toxoplasma gondii, it gets out of host and it can stay in an environment for up to 6 months. Does that make sense? This can stay in the environment up to 6 months. After 6 months, they will not infect anyone. They will not bother anyone. Yes?
Check my understanding. So, however, in the case of toxoplasma, you have to have it in the environment long enough for those nuclear divisions to occur in order for it to be infectious.
1 to 4 days. Yes. Yes. 1 to 4 days. What else on this, guys?
So what's the difference between that and existent?
Okay, so when this is ingested, let's assume, let's talk about this in case of Toxoplasma gondii. When this is ingested and goes into our gut, it comes out of the oocyst stage and becomes sporozoites the sporozoites eventually become tachyzoites. So when they get out of that shell, then it's called excisment.
Okay.
They have to get out. These 8 will get out. And that would be when they get out of this shell, that's called the name of the process is called excystment.
So like an oocyst, would that be like, okay, so you have the sporozoite in there and all the stuff in there, right?
Right. So encystment occurs when in the gut of cat.
Right.
So encystment is occurring in the gut of cat. And when they come in the environment, they're just like a cyst, they are acting like that except the nuclear division occurs.
Okay.
Keeps occurring, but it stops.
Right.
It doesn't keep going and going and again. This one becomes 2. No, it doesn't. This doesn't happen
Okay.
in the environment. Yes.
And in the case of a pregnant mother, the tachyzoid stage is the one that can cross the placenta to the baby.
Right.
And
They
then,
will.
in the case of someone that isn't pregnant, the immune response is what triggers it to enter the brachyzoid stage, where it becomes
Right.
an infection.
Right. But in case of Toxoplasma gondii, we are jumping around all over you guys because of this concept. But in case of Toxoplasma gondii, what happens? When you ingest it first at the beginning, for the first time, the oocyst, or getting it from pork, whatever, goat's milk, from bradyzoids, they break open to tachyzoids, not necessarily from oocyst. From bradyzoids, they break tachyzoids, our immune system slows it down and become bradyzoids in our brain cells, liver cells, muscles, all over our body. And then they break again to tachyzoids, and then our immune system comes in and slows it down to bradyzoids, tachyzoids, bradyzoids, tachyzoids. Natalie, is that right? Bradyzoids, bradyzoids, forever, until we die, which is 120 years. Yes?
So within the oocyst is the sporocytes, right?
Mhm.
And then, so when we swallow the oocyst, the oocyst would break, letting
Very
the
good.
sporocytes-
Undergo excissment.
Okay.
Do it make sense now? Yes. Yes. What else? Who else? Yes, Jocelyn.
What explanation do
You
you
changed
want?
your name to Gap today?
No.
No. Jocelyn still your name?
Yeah.
I thought you said Gap. Gap
What
today.
information do you want for the question?
What?
What is the differentiation of
I
the
cannot hear.
brain?
Microphone, do you want?
What is the differentiation of brain?
Oh, differentiate. Let me ask this question first. The differentiation brain, again, I don't remember it all, but you have to know the reason I gave it to you guys. Did I ask any quiz questions? I might ask.
No, but you said you were
You've asked a
going
bunch
to.
of questions in the lecture on the slides about it.
But remember, one thing becomes another thing in the brain, and that's why I gave it to you guys. So what happens? You have the sperm, right? Enters the egg. The name of the process is called fertilization. Is that right?
Yeah.
And then what happens? This is called zygote with one nucleus. Zygote undergo 2, 4, 6, 8, same size. And then the ball is full of cells, and mitosis is occurring. We call it cleavage. So this is morula. And they keep dividing and dividing, same size. And then what happens? This is called blastocele. Right, seal, and then the stage is blastule. In case of animal, in case of human, it's called blastocyst. And then you will have, I'm not going to draw the cells anymore. You have invagination of the cells. They start folding. When that folding occurs, this area is called blastopore, and that area is called still blastocele. But since you have imagination, this is called gastrula. Right? Does that make sense so far? And then this stage undergo, that was a quiz question today, undergo neurula. neural stage with a neural plate, and after that you have embryo, and then you have fetus. During this one, you have organogenesis, and then finally you have growth.
So, I have a question. On the quiz question, this is like kind of related to the quiz question today. So, neurula is the name of the phase, and neurogenesis is what's actually happening in that phase?
Would you accept
Neurogenesis?
Yeah.
Yeah.
That was on your slide.
You put neurogenesis,
Because you put organogenesis, and then
Okay.
you put neurogenesis.
Yeah,
Are they the
if
same
you write
thing?
down neurogenesis is what stage, the name of the. See, neurogenesis is the name of the process.
Okay, that's what I
The
was.
neurula is the name of the stage.
Okay.
And the name of the structure there was neural plate.
Yeah.
No points.
There was so many.
You just met.
Okay, Nick, is this good?
Yeah.
Okay, as far as the brain goes, you remember, I can't remember, if I have the slide, then I can talk about it. You have it 3, 5, 10, something like that. You have 3 prosencephalon, mesencephalon, diencephalon, and then the mesencephalon becomes 2 things, and then prosencephalon becomes 2 things, and each one of those become a pineal gland, a cerebellum, cerebrum, thalamus, all of those. What is the, if you ask me a specific question, then I can maybe answer it, Jocelyn.
Oh,
What
it was
is,
just in the, on the exam question.
I know, but what is it that is, one becomes another thing. It's just like here, we have blastula becomes gastrula becomes neurula. So I'm saying prosencephalon becomes those two things become pineal gland, thalamus, so on and so forth. That's it. Yes.
What's nisogamy and anisogamy?
What is anisogamy? It means and in front of a word it means not, right?
Right.
So when the cells are not, the gametes are not equal size. Isogamy, it means when the gametes are equal size. In case of human, our gametes are not equal size. Human egg is about 1,000 times bigger than human sperm. Yes?
So is coelom just a lumen in development stages?
It's not a lumen. Lumen, you refer to lumen when you have a tube, any tube, which inside of it is hollow. And material can pass through it, like your blood vessel, your digestive tract. These all have lumen, okay? But coelom, later on you will see it like your heart, okay? So the heart actually, it came from mesoderm, right? There is a sac outside of heart which is called pericardium. I never talked about it yet. Okay, so but there is a space between the sac around the heart and the heart itself. That space is called coelom. And both pericardium and heart came from mesoderm. And there is a space between them. And that space is coelom. So we are coelomate animals. Later, in the next exam, you will learn what is acoelomate animal. And exam number 4, we'll talk about pseudocoelomate and eocoelomate, which we human are eocoelomate. So I'm not talking about that yet. We will talk about it as semester goes on.
What are the different?
She raised it.
Oh, so still they just fill it with this right there, right?
For now, you should know coelom is a space that is just space for now. Later on, you will learn coelom is a space that came from 2 layers of mesoderm, that is surrounded by mesoderm. Later on, we'll learn that. We'll talk about it a little bit later on. Yes? He had a question earlier. Ruben?
Hold up. What are the different modes of locomotion in Batista?
I only mentioned 3. pseudopod, cilia, flagella, 4 of them. Cilia, flagella, but cilia and flagella move the same way, 9 plus 2. And then you have gliding,
Okay.
4 of them. Yes?
Could you explain regulative and mosaic cleavage?
Okay, what happens if what is it that is not clear? Maybe I can tackle that bit.
Probably the differences between each form of cleavage at which point in which angle.
Okay, what happens if you grab a zygote, they both have zygote. Zygote, right? Zygote becomes what? 2 cells, 4 cells. Let's say we stop at 4 cells. In the 4 cells, you separate them, right? Now I'm making some sense, and they all have nucleus. If this becomes deformative, and possibly this becomes normal, okay? Deformative. What kind of, in animal kingdom, what is that? Is that mosaic or regulative?
Mosaic.
This is called mosaic. Right? But regulative, what happens, you zygote, same thing, same zygote, different animal though. It cannot happen the same, different animal. Then you have 4 cells, just like the other one, and then you separate. You go to the laboratory on the dissecting scope, you separate them, and then each one of these cells become a normal animal. All of them. I'm not gonna write it
So,
down. So that would be a regulative.
on mosaic, so they don't all have to become defective in order to be mosaic? So you
Again,
can have
depends
some
on the
normal-
animal. I
Okay.
said defective. Some of them can become normal, but
Okay.
yes,
It just
all
depends on what
it
organism you're
depends.
using? Okay.
But the main thing that you should know, I think your textbook talks about it, I didn't talk about it, because what happens early on in some species, these cells are communicating with each other. And when you separate them, there is no communication, so the cells become defective. Here, in these guys, they're saying, scientists are saying, well, the amount of communication between these cells are very limited. But as you know, if this becomes like a blastula and you do this, it will not. It will become deformative because then there is a complete set of communication. In case of the cloning and embryonic cloning, that is proven. I mean, they're working on it right now. Those communication, what kind of chemical it is that this cell release and that cell gets it. This cell has the receptor, this cell has the, they're working on it very hard. This cell releases the chemical molecule, this cell has, they're communicating at that stage, at blastula. But early on, very early on, maybe 4 cells, you separate them, you get 4 normal organs.
Would a good example of that be starfish?
Starfish, yeah, there are many, in your textbook or lab, or my lecture notes, I gave you, yes, examples of it, yes. Yes. Did I answer, is that okay?
Yes.
Yes, ma'am.
On the question of feeding, one of the questions is, why are bubbles so special to
Yeah, I didn't answer that question, that's a good question. That's a very important question, that's something we are gonna, at the beginning of the material for next exam, that's something, definitely it will be addressed, but right now, Volvox, it seems to be, he's gonna go and carry on with me, but please, don't carry on with me, because that will leave that material for a next exam. Because it's been shown that Volvox was the beginning of the multicellular organisms. You know, we have about 200 different types of cells in our body. Most species have been studied, they look at them, how many different types of cells have. We have 200 some odd different types of cells in our body. So Volvox, based on evolution, and we'll talk about it, most scientists agree that was our great ancestor. So it's a beginning, Volvox are the beginning of multicellular organisms on planet Earth. That was the beginning of becoming a multicellular organism. Just one sentence, it's the answer to that question. Beginning of multicellular organism. Multicellular, Volvox is not a multicellular, Volvox is a colony, remember that?
Yeah.
And a colony means the cells are pretty much same, almost same, like a bacterial colony. You have a bacterial colony, the cells are the same. But when you talk about multicellular organism like us, our intestinal cells, you saw it, is columnar, and our kidney cells are cuboidal, and our fat cells are round. So these, our muscle cells are elongated, and they are totally, their genetic makeup is same, but their chemical makeup of each cell is completely different. And as I said, there are 200 different cells in us. 200 cells. Any other question? Yes?
Can you talk about the animal cloning? Like on the quiz the other day, the answer was white blood cells. It's an animal cloning. They put nucleus in each of the following cells into an empty egg. Like, I got that one wrong there. Can you explain?
I thought it was you put the nucleus of the empty animal.
This one was.
Is that
Then
Yeah.
Oh,
I've been known to make mistakes on those. In animal cloning, they put nucleus of each of the following cells into an empty egg. White blood cells, nucleus. Red blood cells do not have nucleus. Sperm does not have all 46 chromosomes. Egg of another animal, no, they
So
don't
it
do
just
that.
has to
none
be
of them.
any diploid
So
nucleus?
white blood cells has all 46 chromosomes, right?
So can it be just like any diploid nucleus? Like
Any.
Okay.
Any. In case of Dolly, they took the mammary cells of another sheep. Any cell, liver cell, fat cell, as long as the cell has all 46 chromosomes. And that's what they call it Dolly. Is that I didn't talk about Dolly Parton? James, do you know who is Dolly Parton? The country singer in Tennessee.
Yeah.
You don't know.
Will and I do.
What's up?
He has
Dolly Parton.
Dolly Parton. She's famous not only for her singing and her act and everything
Oh,
else,
she has
but
like a
also
book club,
famous
right?
for her breasts. I'm being recorded and being put on YouTube.
She's famous for her.
And this is a true story. The scientist who put his the Dolly because they followed Dolly Parton's breast. They took it.